Okay, kiddos, we're going to do a lab today called Comparing Animal and Plant Cells.
So you've done some stuff already to learn a little bit about animal and plant cells. Now, today is the day where you are going to actually see it firsthand.
So your purpose is on the board, or on the screen here, to compare the structures of a typical plant cell, which we're using Elodea. I've also heard it called Elodia. I say Elodea, but I pronounce words incorrectly anyway. And we're going to actually use a human cell as our animal, because you are a human. Elodia or Elodea is a aquatic plant from like the pet stores where we buy ours. So it looks like this in the corner, in the bottom left-hand corner here. We don't have any to show you, but it's right there on the screen. Okay. So your materials today.
Make sure before we go into materials, though, make sure you have a yellow lab sheet. And you also, there should be a sheet for you to record your data in the crate. And it'll have circles on it for you to draw what you see and things like that. So make sure you have that stuff with you.
Okay, so you're going to use your microscope, your methylene blue stain. You need to ask the teacher where we keep it, because it's not going to stay out on the counter all the time.
Pay attention to the word stain. It does stain, so do not get it on you. Be very, very careful with it.
Right. You'll use a toothpick. You'll need, look at this, it says a leaf. So please don't dismember our plant. You can take the leaf from the top and make sure it's just green and healthy.
And I would use the forceps or tweezers that you used in your letter E lab to just pluck one leaf off of the stem.
Right. And then the rest of the supplies are pretty self-explanatory. Paper towel, two slides, dropper, water, and cover slips. Okay, here's your procedures, and they are written in your handy-dandy instructions here, but we kind of want to go over them because this is a kind of a difficult lab. Not hard, just a lot of steps. So you're going to make your slide with your cheek cell first, and you're going to use your cheek, and the cheek is in the inside of your mouth. I mean, it's this, but we're concerned about the inside. And so you're going to put the drop of water in your slide. Make sure you clean the slide, because again, these are slides we've used year after year, so you don't want someone else's goobers on your slide. And then using the flat end of a toothpick, some ends are pokey, some ends are flat, you'll gently scrape the inside of your cheek. So you take it inside, and look, you can't see anything, but I touched the inside of my cheek. They're there. They're microscopic, which is why we're using a microscope.
And do a couple swipes on your cheek. But really be gentle. Don't come to us and say that you stabbed yourself in the cheek, because we will have no sympathy for you.
And you should not see anything. If you gouged out enough skin that's visible, then you need to be nicer to yourself. Love yourself. And then we have an imaginary drop here. You'll take this, swirly, swirly, swirly. You just transfer them. That's all it takes.
Yep. Okay, so before you put your cover slip on, this is where you're going to use the methylene blue. So you're going to put one drop of methylene blue directly on your water droplet that now has your cheek cells in it. Do not touch the dropper, though, to the drop that's already on your slide. Just do it slightly above it.
Squeeze.
One small drop, okay? It'll mix together on your own, so you do not need to do any mixing. And then you will take your cover slip, that thin plastic square, you will start it at an angle, slowly lower it, and then you will drop it to try to remove all the air bubbles from it. If you have air bubbles around the edge, that's okay. You can use your end of a pencil or your finger if you're very gentle. If you have a air bubble, though, that's sitting right over the center of your cover slip and you can't seem to get rid of it, you may need to remove your cover slip and create a new slide.
Yeah. Okay, this next step is very tricky. If you look at your slide, and we don't have our methylene blue here, if you look at it, it's really, really blue. It's really dark blue, and it's going to be so dark it's going to be hard for the light to go through it. It's almost opaque, if you remember that word. So you need to remove some of that stain. That stain is to actually, the goal is to stain your cheek cells and make them blue. So this is kind of weird. You're going to put some drops on one side of your slide, and then you're going to use a paper towel, bada bing, and put it on the other side of your slide. Sorry. So if I put a drop of water over here, I'll put my paper towel on the other side. It's so cool. It sucks it over. And you'll see that stain escaping. If it's not, you need to put another drop of water there. So if you look at the PowerPoint, there's water here. About 2 drops usually works. is touching your cover slip. You put your paper towel here, it will suck it over, and that stain will then be replaced with clean water. But you're like, how am I going to see my cells? They're stained. They have trapped that blue dye in. And you can't tell because, again, they're microscopic.
Yep, so don't be concerned if you think that all of a sudden, oh my gosh, my liquid is completely clear now. No, it's not. Give it a chance, put on your microscope, see what you can see.
And if it's completely clear, you've done a really good job replacing that stain with the water, so way to go. Okay, next up, same as your letter E. You're going to use your microscope. You start with the low power, always, just to find it. You're looking for the blue dots. And then you go into the medium power, zooming in. You have to find it. And then you're ready, once you have it isolated, then look in the high power. Again, the microscope is always the same. You start with the stage as, well, you put it on the stage, and then you raise it up, and then you lower the stage as you're looking for your object.
Mm-hmm. And you may need to move your slide around a little bit to locate your cheek cells, but once you find them, they should be pretty evident. They should look like little blobs. Blue blobs. And if you notice in number 8, we tell you a couple things that we want you to be looking for. Those are the 3 things that we think you should be able to see. So look for them, fill in your table, and then give us a drawing. Make sure you label those items, draw it to the best of your ability, and make sure you record the magnification. So you learn how to figure out the magnification based on what lens or what power of lens you are on. So write your power of magnification next to your drawing.
You got to do math to figure that out. Make sure you do your math. Okay, and then you're going to clean and dry your slide because now you have your goobers on it and someone else is going to touch it later. So clean it, please. And remember, when you dry, you'll have to clean both pieces, cover slip, and you don't scratch it, you just blot again. Right. Next.
Really, really important too that you make sure that you're putting anything back once you're done with it.
Thank you. Yeah. And then now we're done with the animal. We're looking to the plant. Oh, that's what you're going to be drawing. So this is located on your white sheet. Make sure you keep in mind that I'm asking for several cheek cells, and we want only one plant cell later. But anyway, so the procedures for the plant cell, you'll get your leaf, and you're going to put this guy under the low power, and he's easy to make. You get your leaf, drop of water, done. Why don't we need to stain the leaf?
Because a leaf is a plant cell, and so your plant cells should already have a greenish tint to them because they have these wonderful things called chloroplasts.
That's right. So you're going to look for the Eladia cells, find it under low, then go to medium, then high, and you're going to find a few things: the cell wall, the chloroplast, and nucleus, of course, in the cytoplasm.
Mm-hmm. So you'll see some things that you already saw in the animal cell. But you'll see some things that you did not see in the animal cell.
And then when you're done, you'll clean and dry your slide and cover slip. So here's a sneak peek. I know I hate seeing the ending of the movie before the movie comes out, but you need to know what you're looking for. A lot of these dots, they're just goobers. I don't know, they're goobers, they're dirt, maybe they're breakfast, don't know. You're looking for these doohickeys here. Sometimes the doohickeys are folded, or clustered together. Sometimes they're on top of each other like these guys. These are cells. There's 3 in a row here. But try to find those. And the rest of these, just ignore. There's just debris. And then your Eladio cells, they look like bricks. You can mess with that magnification and you'll see varying thicknesses and stuff. So try to get it because you can tell there's something blurry underneath it. Try to focus on this kind of object. All right.
So this is the chart that you will fill out as you go. So you should have your parts for your cheek cell and your parts for your Eladia cell checked. Not every single box should be checked for each cell. Pay attention to the type of cell that you're working with.
And finally, you do have some analysis and conclusion. So answer the questions provided. They're on the white sheet. And please use complete sentences. Avoid the pronouns like it and they. We need to start using like actual nouns. Be specific of what you see.
So instead of saying in the cell, okay, well, what kind of cell? In the plant cell, it had a dark spot in the middle. What had a dark spot in the middle? Be very specific with your answers.
And then here's your critical thinking. Do the best you can to answer those in complete sentences also. And if you have any questions, sometimes the kids kind of confuse them. Especially question 3, that confuses kids typically. Seek help.
Make sure you clean up all your supplies, okay? With great power comes great responsibility.
Thank you, Spidey.
You're welcome.
Okay, that's the end of it. If you have any questions, come see us. Later, Skater.
See ya.